In this video, I'm going to be talking about Watson and Crick's model of DNA. So DNA is made up of strands of nucleotides connected together. Nucleotides consist of phosphate backbone, sugar, and base. And these strands are twisted into a double helix shape due to the hydrophobic interactions. This is an example of base stacking. As you can see, the bases are paired together, but they're also stacked up, kind of like a ladder. And then these bases, these strands, are twisted into a double helix form. More specifically, this is the sugar of the DNA, and as you can see, the 1' carbon is connected to the base pairs. The 2' carbon distinguishes between RNA and DNA, so RNA would have an OH group connected to the 2' carbon, but since this is DNA, it does not have that. And then the 3' carbon and the 5' carbons are connected to the phosphate backbone. As you can see, the 3' carbon of this sugar is connected to the same phosphate backbone that the 5' carbon of this next sugar is connected to. So it creates this long chain of strands. There are 4 bases: adenine, thymine, cytosine, and guanine, and these are characterized into purines and pyrimidines. So purines, A and G, have 2 rings, while pyrimidines, C and T, have only 1 ring. And base pairing is present in DNA, which means that adenine and thymine are always paired together, and cytosine and guanine are always paired together. And this is an example showing how, for example, thymine and guanine would never pair, but guanine and cytosine and thymine and adenine fit together perfectly and will always pair together like puzzle pieces. DNA also has anti-parallel strands, which means that the strands are connected in a parallel manner, but the 3' ends match with the 5' ends. So as you can see here, when 3' is connected with 3' and 5' is connected with 5', they just don't fit. But when 3' is connected to the 5', it fits perfectly. And so this makes the DNA strands anti-parallel backwards. So what makes the base pair stay together? The first way is the hydrogen bonds. So A and T are connected by 2 hydrogen bonds, while C and G are connected with 3 hydrogen bonds. But these bonds are not very strong. What's actually stronger in keeping the base pairs together are the hydrophobic interactions. So bases are hydrophobic, while the phosphate backbone is hydrophilic, and the surrounding makes the hydrophobic bases stick together and twist into a helix shape. Watson and Crick's model has 10 base pairs per twist and is right-handed. You can see the difference between left-handed and right-handed. The accepted model is right-handed, so it twists up and to the right. There are major and minor grooves in DNA. Major grooves are the areas that are wider in the twist, and minor grooves have less area in the twists. When DNA is placed in an SSC buffer, it causes the negative charges of the phosphate backbone to neutralize, which keeps the DNA more stabilized and able to stick together. This causes the TM of the DNA to raise. The TM is the temperature that is required to denature 50% of the DNA. And denature means to separate the double strands into 2 single strands. And because the bonds are harder to separate, the TM is raised. These are my references. Thank you for watching.